So, hello everyone. We are into episode number 3 of Clinical Research Talk series. So, in this particular session, we are going to talk about clinical data management and analysis. So, as we know that data is the next oil, and it is very important that we know how the data is collected, especially when it comes to healthcare data, the clinical data. Then it is very important to understand that what clinical data management is, and once the data is collected, how that data is analyzed. Okay, so this session is very interesting and critical to understand the concept of clinical data and how the analysis is carried out. So let's start this session. So before we start, I would like to inform you that this particular series is in collaboration in between 3 YouTube channel. So first one is Alpha to Omega, which is run by Aditi. So here you would actually find the concepts of data analysis, how the data is handled, because she is extensively working in it. And the other two channels, Clinical Aim Research and Clinical Research X360. So these particular channel will essentially give you an idea how what is clinical research and what are all the activities related to it, and also what are the updates into it. So without any further delay, let's start this video. So first we'll talk about what clinical data management is. So we must understand that clinical data management is the entire process of management and storage of data in a clinical trial. So when you conduct a clinical trial, your primary motive is to find out if a particular medication is effective or not. But that is a very rudimentary explanation. What do we mean when a particular medication is effective or not? We mean that does the scientific data support for a medication to be effective or not? And we do not have to verbally convey that a medication is or a vaccine is effective. We have to scientifically demonstrate it in terms of data, in terms of number. So when it comes to clinical data management, it is a field which deals with the clinical data generated during the clinical trial, as well as the entire aspects of the life cycle of clinical trial. So who sets the standards for clinical data or the data management? So it is done by CDISC, which stands for Clinical Data Interchange Standard Consortium. So this particular agency sets two standard protocol which must be followed while managing the clinical trial data. Okay, so there are two types. First one is Study Data Tabulation Model Implementation Guide for Human Clinical Trial, which puts emphasis on the standard model for organizing and formatting data for human and animal study. Okay, so here you would get guideline or clear-cut understanding that how a particular data and how the data model should be organized and formatted for human and animal study. The second part would be the clinical data acquisition standards harmonization, okay, or C dash. So which essentially helps you to standardize the case report form, because case report forms are something where you collect the data. Okay, so it can be a literal form. Okay, so which essentially what it does is it collects the clinical data. So as you can see here in CRF, what we do is we enter the clinical data, as well as we also electronically capture the data. Okay, and for collection of the data, in a lot of countries which have gone digital, they use digital media. Okay, there they will use the electronic data capture systems. And in India, what we do is we go for paper CRFs, where we enter the data into paper-based CRF and then enter that data into the electronic data capture system. Okay, and what you would get question in your mind is which are the standards? Okay, which are the global standards which clearly highlights the use of that data or how a particular case report form should be. Okay, so when it comes to that, it is regulated by the Code of Federal Regulation or CFR. In CFR, we are particularly talking about 21 CFR Part 11. Okay, so this is a very much thing to remember when handling clinical data management. So 21 CFR Part 11 deals with clinical data management. Okay, now moving on, we are coming to electronic data capture devices. So once you capture data in your online database or in paper CRF, that particular data has to be entered in web-based application. Okay, so you enter that data online and you use that particular system or software which you call EDC or electronic data capture devices. You collect the data and you store the data. Now this is particularly done to collect the data in a uniform format, and once you store the data, that data can be analyzed. Okay, so electronic case report forms, what they do is they essentially collect data related to the study. This data can be patient demographics, the height, weight, the medical history, and any other relevant information which can be effective in proving the efficacy of a medication. Now, please note that whenever the data is collected, the data is already, the case report form is approved by the regulatory authority and the institutional or the site ethics committee. So, when it comes to data privacy, which is a concern nowadays, so only data which is mandatory to prove the efficacy of the medication, only that particular data shall be collected. No unrelated data is collected in these particular systems. Now, what are the some examples of electronic data capture system? So, some famous examples are Oracle Clinical. Okay, that is used very widely. We use Rave, we use eClinical Suite, we use Clinical Trial, we also use Macro. But it is very important to know that there are also some open source electronic data capture system like TrialDB, OpenCDMS, OpenClinica, and Fosco. So these particular system can also be used to build on, and these are open source. So any particular person who is interested in pursuing electronic data capture software development, they can pursue this particular platforms. Okay, and as I've said, the Code of Federal Regulation CFR 21 Part 11. So it not only explains you how the data should be collected and managed, it also provides you with the compliance standard. Okay, when I say compliance standard, it means that not only the structure for the database, but also how that database should be maintained. Okay, and that has to be implemented into this CDM system. Okay, and that adherence only leads to submission of that data to regulatory authorities. Okay, so here you understand that the data is collected physically also, electronic also, and when it comes to electronic data capture, it is stored in a single web-based application. Okay, where that data is entered and that data is pooled and collated, and that data from phase 1 to phase 2 to phase 4, during all the phases, that particular data is then moved forward and analysis is done. Okay, so now moving forward, we must also understand what clinical data processing is. So I would like Aditi to explain us the data processing aspect.
Yeah, thank you, Saurabh. So, data processing is one of the most important parts of the of analyzing data, because when a data, first of all, when a data is analyzed, it is analyzed in a software as a data set. So as we know, when we collect data, it can often be very in simple terms, it can be very messy. There can be missing values, there can be missing there can be numerical, there can be, sorry, non-numerical data. For example, some people can, sometimes when a patient doesn't show up for a visit, or they are not part of that visit, it's often written NA, in like not applicable. So that we basically, when we want to clean data, in simple terms, we want to remove non-numerical data, inconsistent data, which is resolved in the form of queries, and missing values. And another part of data processing is, after we are done cleaning the data, we have a data set which is clean, which does not have non-numerical values, does not have missing values. uh, we further analyze it and we take the summary statistics. So summary statistics are basic mean, median, maximum, standard deviation range of the data. And we also visually represent the data to understand the trends and the demographics. So in this visual representation, you can look at different options, like say take a bar chart or a box plot or a scatter plot, depending on what you want to see. So for example, if you want to see a range of data, so oftentimes it might be really helpful to take a box plot. But the same thing is not applicable when you want to see the concentration of the drug versus the time. So you really have to make informed decisions on what kind of visual data you want. And the summary statistics will be greatly helpful to guide us further in when we want to analyze the data as well as when we want to visually represent it. So after this initial pre-processing of data or processing of data, we take the final data and export it to a statistical software like SAS or SPSS for further analysis. We can also use other open source software like R or such to generate reports, and oftentimes R is already integrated into other statistical softwares. So this is a very important step. So now we get on to the data analysis. So this is after we have done our basic data summary statistics, and we want to really get a statistical value. Now, it is very important to, when you're analyzing data initially, to analyze it to as they were originally randomized. So basically, you try to isolate the group according to what original randomization they had. So, for example, if they were in an active group, or if they were in a placebo control group, you will randomize them, you will analyze them according to their original randomization, and then you will start comparisons. And the type of data analysis or the data analysis methods, they would vary with, uh, I mean, the first thing we would consider with the variation is the primary endpoint. So primary endpoint is a very important mark in how we want to analyze data. And the other important mark is how many endpoints we have, and how much data we have can also really influence our data analysis. Now, in here, primary endpoint, there are roughly two types of primary endpoints. Uh, there could be a continuous endpoint or a discrete endpoint. So what is a continuous and a discrete endpoint? What do these mean? So a discrete endpoint is basically an isolated form of data. For example, if you have a drug for cancer and your endpoint is after say 72 hours, how much tumor reduction has, how much have the tumors reduced, then you would take this as a discrete endpoint or some sort of specific endpoint that you want. Whereas a continuous endpoint could be an endpoint like blood pressure. Like if you take a drug for hypertension, then how, and you analyze from 0 hours to 12 hours and different time intervals, then that would be a continuous endpoint. So then these kind of endpoint would greatly influence our type of data analysis methods. So here are a few examples, like for continuous endpoint, a very appropriate test could be the analysis of variance, or also called as ANOVA. So this will be when you have two or more comparison groups, and you can use ANOVA. And for discrete endpoints where you have a lot of numbers, you can use a null or alternate hypothesis, so or a regression analysis. Now, when we have multiple comparisons, like when we want to compare between different groups, it often can get very tricky, because one of the problems is that as you have multiple groups, you have an increasing number of false positives, and that can also
Yeah.
cause a lot of errors. So here you would use pairwise tests that minimize those errors, like Bonferroni method or Tukey's test. And what happens in big data when there is missing value? Because despite data cleaning, we can still have missing values. We can still have missed visits, and we since it's against the rules to fabricate data. But in statistical analysis, you need to do a little bit of that, because otherwise many programs will find it very difficult to analyze that data. So what you approach to that is that oftentimes the last observation carried forward techniques is used. So this is basically when you see the last value, and you add that in the missing value. But oftentimes this can also cause a lot of bias estimates or incorrect estimates. So it can be really tricky. Sometimes you really have to just make an estimate or an average estimate and add it to the missing value. Sometimes you have to delete the rows or delete where you have missing values, but though you want to avoid that as much as possible. So this can be tricky, and it can be very important. So the softwares that are usually used to analyze uh, clinical data statistically are SAS, SSPS, and R, like I said. So SAS, SSPS are not open source, but whereas R is open source, and they are used for clinical data analysis. Yeah. Now, so we've talked about clinical data analysis. I want to introduce something called data modeling or clinical data modeling. And I want to talk about why specifically we use data modeling and why is it an addition to data analysis. So first of all, what is data modeling? So data modeling is essentially, in this context, when you apply a certain mathematical model or a mathematics based statistical model to come to conclude certain decisions related to clinical trials. Now there are a few types of clinical trial models, and there are pharmacokinetic, pharmacodynamic models. So these are when you separate the pharmacokinetic data and the pharmacodynamic data. So the pharmacokinetic data is based on the absorption, metabolism, distribution, and excretion of a drug. So that is, and the pharmacodynamic data or the pharmacodynamic model is based on the effect. So in pharmacokinetic models, you have the one compartmental model where you can consider the body as a compartment, and or you have two compartment models where you separate two compartments, or you separate them into further compartments. And and pharmacodynamic models are when you model the effect that a drug produces. So there are different kinds of pharmacodynamic models, like an Emax model or a direct effect model, or sometimes a delayed effect model. So this pharmacokinetic pharmacodynamic model is a basic structural model to describe how the drug metabolizes in the body and acts on the final effect that we want. And we can always add to pharmacokinetic pharmacodynamic models, or as I prefer to call them, PKPD models, by doing compartmental analysis. So compartmental analysis is when you get specific data with the PK model. So that is when you get data related to the absorption coefficients, or the volume of distribution, or the excretion, and how it's metabolized. So there you would take the intercompartmental clearance. So these convert the pharmacokinetics into mathematical values. And the pharmacodynamic model, as I said, would model the effects, and you can make them more complicated, or you can when you explore covariates. So covariates are demographic data which could be very important to the effect of the drug. So though you do not count it in your final outcome, but it does affect your final outcome. For example, the example of covariates are weight, height, sex, renal efficiency, and other other properties which can really affect both the pharmacokinetic and indirectly the pharmacodynamics of a drug. And the other examples of the data modeling is physiology-based pharmacokinetic models. So physiology-based pharmacokinetic models are when you actually add elements of physiology to pharmacokinetic models. Like you talk about blood flow in certain species, or liver efficiency, or certain other surface area, or certain other aspects within living beings. So you don't only do physiology based pharmacokinetic models, or as I like to call them in short, PBPK models, on only human beings, but you can also do them on animals, on in the preclinical phase. And PBPK models can be very helpful in understanding certain outcomes. Like when we are going from the preclinical phase to the clinical phase, we can often refer to PBPK models. And drug metabolism and pharmacokinetic models, or DMDPK models, here you understand the pharmacokinetics in relation to drug metabolism. So there, and these data modeling can often be very complex, because you cannot, there's no one size fits all approach in the data modeling, because anti-cancer drugs have different type of models. Each drug can have its own and should have its own individual model, whereas a cardiovascular drug can have a different model. So it really has to be, I mean, the most common approach to data modeling is that one would start with building a basic structural pharmacokinetic model, then add then one would add different kind of variances, and then covariates, and then go on to build a more complex pharmacodynamic model, and then go on to build a PBPK model if required, and a DMPK model if required. So it's most of the time it's done like that. And there are several softwares that can be used for this for data modeling, like NonMem, Monolix, and Berkeley Madonna. And as I said, these models are highly variable, but they're very useful, because now they're even used to make regulatory decisions and give deeper insight into clinical pharmacology. So you can refer to these models for even say dose selection and other kinds of understanding the metabolism better. But I mean, maybe you would have a question to why you would need a data model, or why is it more beneficial than more beneficial than only statistical analysis. And the answer to that is that oftentimes clinical data can be very complicated, and they have a lot of mixed effects which are not linear. So as I mean, the key word here is that non-linear mixed effects can make the data analysis very difficult to come to a statistical decision. And it's very difficult to understand about a drug using only statistics, or only maths, or only say a t-test or an ANOVA. So here you go a little in depth and try to explain the drug in a mathematical way. So this is our chance to really understand different kind of aspects of the drug. And yeah, so this is data modeling. Yeah. So now machine learning. So machine learning has very wide applications in drug development, from the preclinical development to the to drug discovery, to the pre-trial planning and execution, the executing the trial. So machine learning can be used in every part of the trial, from patient identification to recruitment to data analysis and protocol adherence and endpoint selection. So patient identification, because you can identify certain patients based on different kind of description biomarker, perhaps in a database. Though that, I mean, you can use machine learning for the data analysis, And machine learning is strong enough to detect any sort of protocol deviation in an ECRF, and also it can be used for endpoint selection. Now, supervised learning is a machine learning method which makes decisions based on known data. So, for example, makes decisions and predictions based on known data. For example, if one wants to have a clinical trial for, say, an anti-cancer drug, and they want to predict the dose of the anti-cancer drug based on, like, because at each point we will be deciding the dose for the next phase of the trial. So you can use this supervised learning method to predict what would be the next dose for the data of the trial, or make informed decisions based on known data. For example, you can predict side effects or predict how many side effects would happen or what the outcomes are using supervised learning. Now, unsupervised learning is a method which is a machine learning method which generates clusters and patterns within generated data or unknown data. So here, unsupervised learning can be used to understand patterns in, say, each phase, understand if there were like a specific demographic group which responded to a drug in a different way. So unsupervised learning is basically used to generate to find patterns in unknown data, and it also has a big role in the post marketing surveillance of the data, where the data is a lot more heterogeneous. And deep learning can be used in big data analysis, and has a large capacity to process different type of data. Now, deep learning is such a thing that it can even process images, sounds, and other. It's not just written data, it's not just text or numbers, because sometimes when we have clinical trial, often we can look at different kind of outcomes which could be in an image format. For example, if we have an anti-cancer
Or
drug,
in
and
even in vaccine, sorry to interrupt, even in vaccine study, what we do is whenever we administer a vaccine, there are adverse event which are in terms of swelling or in terms of redness. So if even the image data can be processed along with
exactly,
clinical data using deep learning, so it will not
yes,
only use the adverse event rating, it will also use that images of swelling or redness, and it will enhance the ability to perceive the adverse event of a particular
yeah,
medication. So that can be
yeah,
helpful.
yeah. I mean, exactly, like medical data is not just numbers, but it can often
Right.
be images, like as you said, the vaccine trial and cancer trials are often images, because patients have to go through an MRI machine or a CT scan or other kinds of scans, and the data that is generated through them is oftentimes it's an image. So with that, with using deep learning, one can really analyze data using those images. So the deep learning that way is very powerful to it's a very powerful tool to detect patterns, and it could be the next revolution in different kind of trials like cancer and vaccines and cells, and there are many several type of trials that can really use deep learning. And so, in conclusion to whether machine learning is used and what are the limitations, I'll just say that, okay, a lot of analytical softwares have inbuilt machine learning algorithms in them already. So they're already in use. For example, SAS has an inbuilt machine learning algorithm, but there are drawbacks of relying on only machine learning. And one of the drawbacks are that machine learning is, in the end, it is mathematics. It can also give false positives. It can predict incorrect values. So there is a very big problem with machine learning called overfitting. So overfitting can either happen when the data is noisy or not cleaned well, or when the data set is too small, and it's basically when the predicted values of machine learning are very close to the input values. So that can be a very big problem in giving us incorrect predictions. And when it's coming to drug trials, incorrect predictions are pretty dangerous. And that is why, in conclusion, if you could go to the next slide. Yeah. Yeah, so in conclusion, this is one of the reasons why these three are so important for a good outcome, because it is very important to have clean data in order to, I mean, firstly, it's very important to have collected the right data in order to have the data that we need or want. And it is important to process data in the correct way so that we don't have any problems or we don't have incorrect predictions, because it can mean life and death in a clinical trial, because ultimately these drugs are going to be released in the public. So data processing is important to clean the data. And for data analysis, we need to be very open to multiple type of techniques. So it's not just data analytics or machine learning, but a combination of different kind of analysis methods, which is very individual to each trial and each outcomes. So I mean, as I said, a combination of data analytics, mathematical models, and machine learning can help us get the best outcomes that we need, and that is tailored into each clinical trial. And yeah, so I just want, in conclusion to what we discussed today, I would like to talk a little bit about the next upcoming episode. So up next, we are going to be breaking down a clinical trial into different phases, and we are going to be talking about the statistical analysis in different phases, from phase 0, which is a first in man study, to the phase 4, which is the post-marketing analysis. So we're going to try to approach different kind of statistical methods and what kind of statistical methods are applicable to what phase. And we're hoping that our viewers would get an insight into different analytical techniques at each step and why these techniques are important. So thank you for tuning into this session, and we really hope you enjoyed. And as we mentioned, we're really up for suggestions of different kind of videos you want to see, or different kind of
Yeah.
topics you want us to cover. So thank you so much. Yeah.
Yeah, so I mean, thank you, Aditi, for explaining us, because being a clinical research professional from the operations part, what we essentially focus is on subject recruitment and the data generation and entering that data into the electronic data capture systems, okay, ECRFs. But in today's session, it was very helpful to understand what happens to that data. And when it comes to using, I mean, data processing, data analysis, what are the different aspects of it? And most critical, which even I got to learn, was how machine learning can be used, because I always felt that using numbers and particular words or rating of any adverse event or medical data, so that was one aspect of it, and that was very 2D type of analysis. But as you clearly and very critically explained, that there are also data in terms of images, okay, that images can be images based on your adverse event, based on your MRI data, your X-ray data, and finding a pattern which we can significantly prove when it comes to submission to the regulatory authority, then that data can be tremendous. And using the supervised learning, unsupervised learning, and even deep learning to make that data all round, not only comprehend in terms of numerical values or word values, the images can also be integrated, and that data can also predict patterns and help you in analysis of tremendous amount of data in terms of deep learning. So
Yeah.
that I mean, when it comes to overall analysis of a trial or overall data submission, when you say that we have submitted all the data, then data not only terms of numbers and words, the data in terms of image analysis. So that could be the future, you correctly explained, of the clinical trial. And I think machine learning and big data analysis can be the next, what do we say, comprehensive methods of analysis of a clinical trial, because I think data which we provide today is what the regulatory authorities want. But when it comes to image analysis and all kind of pattern prediction, so even in the cancer trials or even in vaccine or other type of trial, if we can use deep learning or machine learning to predict the dosage, we can significantly reduce the number of subjects which are exposed to some drugs which can be carcinogenic. So there, these techniques can be very helpful in providing insight. I don't say use it directly from the word go, but to get a critical insight of
Yeah.
deciding a dose. So
Yeah.
these techniques can really revolutionize how we understand clinical trials today.
Yeah, I mean, and as I often mention in different kinds of fields, that machine learning is not a tool to replace people, but it's a tool to kind of make people's understanding or the understanding of the world better. And in terms of clinical trials, it's definitely true because machine learning will really open up the world in terms of like from the when you're in the drug discovery phase itself, when you look at structural data of different kind of molecules, or maybe when you do the cell like the in vitro tests, you look at different kind of images, then itself like machine learning can start, you can use machine learning to process images as long as there's that much data which is clean. So that is one of the drawbacks in machine learning, is that you need that clean data. I mean, a clean data to get a good conclusion. And then you can use it in the whole clinical trial and the post marketing surveillance. So post marketing surveillance, it could be such that okay, there's like a certain side effect detected in a certain population, like say if there's a certain side effect that are happening in women, for example, or women of a certain age group, like they see that okay, younger women are suffering from this more, then machine learning is capable of detecting that pattern. But then as I would like to also kind of highlight that, you know I know these are very fancy words, but they're not the end all of clinical data analysis, because um, it's really important to have a combination of everything, because
Correct,
if you want to have the right conclusion, if you want to have the right data, because since we are dealing with human lives and we're dealing with life and death, it's really important to come to the right conclusion statistically, and in a correct way. So which is why we look at combining everything, combining different kind of statistical models. And yeah, I was really happy that we got to have this discussion with each other.
correct, correct. And also, it would really help the viewers too, because you can really get information how the clinical trial is conducted anywhere. But what happens to the data? Because at the end of the day, when you go to regulatory authorities to get your approval, they essentially will ask you the statistical significance of that particular data
Yeah.
in terms of your vaccine or the medication.
Yeah.
So if there are techniques which are helping me to analyze that data or at least predict a pattern, then I can do two things with it. Firstly, I can reduce the risk. Okay, so when it comes to data prediction through deep learning or the dose decision to have a dose that is to be tested. So if I get an idea, at least I don't say we can go to conclusion on the deep learning itself. If I get an idea or a pattern, then it helps me comprehend the data itself primarily. And the
Yeah.
second thing would be that once, I mean, be it any field, you move from one technique to another. So these can also be stepping stones of data analysis of the future,
Yeah.
because as we move forward, the data is going to get huge. There will be modification in CFR part 12. Okay, code of federal regulation will also accommodate such analysis method, and until and unless we work on various techniques, we are not going to get to that particular system where it will be acceptable to the regulatory authorities as well. So it is a work in progress, I agree. But if someone is helping me to analyze a particular data or helping me to predict patterns, then it is very good for my understanding as well, where my clinical trial is heading, what is the meaning of my data.
Yeah.
So this is a very exciting field to look in, and I would suggest our viewers and listeners to really pay attention in this field also. So it is not only just data entering into the ECRF, but what happens to that data. That is also a critical part, because essentially you will get approval through that too. So it is a very good discussion to have.
Yeah, yeah, and at each step the outcomes are really different, because you can I mean you have, as I mentioned, that anti-cancer drugs have a different kind of way of analysis. I mean, it's really entirely dependent on the outcomes and what you really want to see. So it's not that if I used this data analysis method in a say in a vaccine trial, then I can't use the same thing in anti-cancer drug. And this is why it's really important to not just do statistical analysis on SAS or something, but also to really look into it,
Correct.
integrate other techniques into it,
Yeah,
because there's
and
no
also
one
I would
size. Yeah.
yeah. Also, I would like to add that that is the thing that we are going to see in our next session, that what happens in one particular phase of trial and how one phase data analysis or statistical analysis differs from the another. What happens during the each and every phase of the trial. So that is going to be our next session. So we are up for another interesting topic.
Yeah, and this
Yeah,
is, of course, this is the bibliography without which I don't think we would have had such good content. So I really thank
of
all
course, of
those
course.
people who put their articles out there, and I hope it will encourage more people to publish what their findings are, be it in their education or some other time. So because it really helps, and sharing is caring. So in
Of course, of
conclusion,
course.
we hope to see all of you in our next session, which is phase analysis of phase trials, and we
Yeah,
hope you enjoyed this session. So thank you so much from my side again. Yeah.
yeah. Thank you, everyone.
Yeah.